what is DNA? DNA is just a really long molecule that contains the unique genetic code of an organism. Essentially, it's like a giant recipe book, like a cookbook or like an instruction manual for building an organism. It has all the instructions of how to build a human or how to build a mushroom. The sort of unit of measurement in a sort of string of DNA is a base pair. A basidiomycete will typically have around 47 million base pairs. A human will have about 3 billion base pairs. So these are pretty long strands of DNA that we're talking about. That's got all the information on that organism. But the good news about fungal sequencing is like we don't have to look at that whole gigantic string of DNA. We're actually only looking at a tiny, tiny region. It's sort of like one page of the encyclopedia. And that tiny region is called the ITS region. So what's the ITS region? It's basically a completely useless part of the DNA. It connects two really important regions. It's a spacer DNA. And when something goes wrong in these important pieces of DNA, like a mutation, because mutations happen all the time, that's really bad. Like the organism will, either die or it won't be able to reproduce, or it'll have to somehow fix that mutation. But when a little bit of that spacer DNA, when there's a mutation on that, nothing happens, the organism doesn't die. In fact, nobody cares. So what happens to that bit of spacer DNA, that's about 700 base pairs long, is like it accumulates mutations. And those mutations sort of reveal essentially how similar, how different two kinds of fungi are. If the pattern of mutation is almost identical, that means you have very similar specimens. Or if the sort of pattern of mutation is very dissimilar, you probably are not very closely related. You're probably very far apart on the fungal tree of life. So it's a really useful little section, and the discovery of this little spacer DNA has been sort of a revolution for mycology, because that allows, by analyzing that little piece of DNA, we can now sort of compare one mushroom to another and figure out what is the same species and what isn't. So, so that's what DNA is, and that's what fungal sequencing is. But why do we do it? That's sort of one of the questions. And obviously, when I ask people, what's the point of sequencing DNA? They say like, well, to get the names, because we all have this innate sort of desire to get a name, to put a name on a specimen. We've all been, at the foray table and we figured everything out, but there's always like one or two specimens that we cannot, by the best will identify, and we go home, we hit the books, we hit the microscope, and we still can't figure it out. And then our curiosity becomes so intense, we go like, maybe DNA sequencing is going to give us that name. And obviously DNA sequencing can do that, but it also does a whole lot more. It's sort of an absolutely fundamental part of mycology these days. Because if you don't have clearly defined species, you can't really study them. You can't analyze their pharmaceutical properties, their medicinal properties, their ecological properties, because you're not really sure what species you're talking about. So what taxonomists do, sort of as a matter of course, is looking at the DNA sequences of species and try and sort of organize them. This is just an example, sort of I picked out of a paper I was reading. Elsa Wellinger is a giant in the Lepiotaceae, and she kept finding this little Lepiota, and it had a name. It was called Lepiota oculata. It was described in the 1930s by Zeller, but she kept finding similar looking specimens that weren't quite the same. So she sequenced the type, and then she sequenced a whole bunch of her own collections, and the DNA kind of showed that instead of falling into a single bucket, like, as you would expect, they actually fell into 4 fairly separate buckets. And then what a good taxonomist does, they start looking for a macroscopic and microscopic differences, because you don't just want the DNA sequence to be different. You want to find a clue that you can use in real life. And she found microscopic and macroscopic differences. And in fact, it turned out to be not just one species, but 4 species, which she named. In fact, 3 of them were Leucoagaricus. They weren't even Lepiodas. And then she wrote a paper about it. And, and now anybody who wants to study those species, they can just go to that paper and it's all there for them. And taxonomists are completely undersung sort of heroes of mycology, because without taxonomy, nothing else can really work. They tend to be underfunded. It's not breakthrough science. It's not considered sexy. So, there's not enough taxonomists, and the taxonomists that they are, aren't able to do enough field work. So there's definitely a shortage of sort of taxonomic expertise that's out there in the world. And obviously that's, an opportunity potentially for people like us. But that's how scientists use sequencing. It's obviously revealed a lot of different things about the relationship of fungi. In the olden days, people used to group fungi together by macroscopic characteristics. For example, tooth fungi. It's a term of convenience that, in fact, we still often use for things like the Bancuraceae and Oriscalpium and Hericium. They're all toothed. But do they really share a lot in common beyond the teeth? I mean, sequencing quite a while ago revealed that actually some of them do, some of them don't. It turns out that the Russulales include not just Russulus and Lactarius, but also Oriscalpium and Albatrellus and Bundersevia and things that don't look anything like each other and don't on the surface seem at all similar, turn all out to be part of the order Russulales. The sequencing showed that somewhere in the not too distant past, they actually shared a common ancestor, and we can only speculate at the evolutionary pressures that made some of them have gills, some of them spines, and some of them pores. So that's sort of one of the big discoveries from genetic sequencing. The other thing that sequencing has helped us do is sort of unlump species that used to get lumped together. And that's sort of a bit of a historical accident in that mycology was developed in Europe. So when American mycologists started calling things name over here, they used a lot of European names. And sometimes, indeed, that European name was correct, but oftentimes it wasn't. In fact, Rolf Singes speculated that up to 90% of American species are actually different species than what we find in Europe and obviously other continents. I mean, just to use one example out of, a million, Amanita rubescens, up until very recently, was a species that we thought we had in America. Turns out what we have here is something called Amanita amerirubescens, and it's actually a complex. It's not a single species. Rotellus has done a lot of work on that. And something else that on the West Coast that we used to think was also Amanita rubescens turned out to be Amanita novinupta. They all have slight morphological differences. Amerirubescens is a little shorter and often a little yellower in the universal veil. Amanita novinupta is completely white in its sort of larval early stage. So there's usually sort of a little tell that tells you it's a different species, but it's not always to pick them up unless you know that there's something to look for. So it helps us split, I guess. The other thing it's been helping us do, and some people welcome this and some people don't, it actually has led to genera being split. And I mentioned Cordinarius. The Inocybaceae is another example. Back, I think in 2018, Brandon Matheny looked at a lot of sequences for what used to be called Inocybe and found that they actually fell into 4 distinct buckets: Inocybe, Pseudosperma, Enosperma, and Malocybe. And they were quite different from each other in terms of the presence and absence of Calycystidia, of spore shapes, of whether they had certain kind of veil, certain kinds of cap characteristics, and so on. And he split them, and it's now 4 different genera. And we have sequencing to thank for that. And some people are still a little bitter about it. It's mostly been accepted, but not everybody's happy with it. But once again, just looking at what clusters together genetically has led to this kind of outcome. So what sequencing then really does is it allows us to sort of map out the relationships between fungi, between all fungi really. It allows us to build this gigantic family tree. It's a sort of beautiful graphic that showed up in Nature magazine a few years ago, which is essentially a family tree for all the different groups of fungi that there are. And you can see that some of them are really old. Some of them sort of have these green lines that go all the way to the Permian and beyond. Whereas some other ones, like the Cantharillalis and the Dacromycetes, and so on, those are really, really old kind of, I guess, families. And then, as you go around the tree, they get younger and younger, and you kind of end up with the Inocybaceae and the Strophariaceae that are, in the like in the Paleogene, like they're much younger, so they split from the ancestors much more recently. So it's it's led to this beautiful understanding of how all fungi are related, and it showed us that many traits and characteristics have evolved over and over again and show up in many different ways in many different kinds of taxa. So it's been kind of an amazing thing for mycology to have these techniques that do that, but that's not all. The other thing is, this doesn't have anything to do with sequencing yet. Longitudinal studies reveal that there's just a tremendous amount of fungal biodiversity in the world, even in small areas. These are two landmark studies, one at Observatory Hill by the Cheskas in Victoria, BC, one in La Chanias in Switzerland. Both surveyed really small areas, like 200 acres or less. That's like a quarter of Central Park, and found, up to and over 1,000 different species in just these tiny little areas. So we know that there's just a ton of fungal biodiversity all around us. But the challenge is, finding it, discovering it, describing it. And that's where sequencing kind of can help. What these two charts are, they're so-called species accumulation charts. The first one, the one on the left, maps the number of species that have been found in Nanopore since the 1960s. And as you can see, the line goes up and up and up, and there's just no sign of it kind of plateauing. Essentially what it tells us, the more we keep looking, the more we keep finding, which is kind of crazy. Right now, when the chart was done, and that was like, that was like 15 years ago, there were at 2,858 fungal names, and I'm sure they're well beyond 3,500 at this point. A very similar curve occurred when Stephen Russell was looking at Russula species from his Indiana collections. Garrett Taylor recently did his own species accumulation curve. It had exactly the same shape. I saw one for Newfoundland. So basically, wherever you look, the curve looks exactly the same. The more we look, the more species we find, and there's really no sort of foreseeable end to it. But to really, find all this diversity and document it, so sequencing is an incredibly helpful tool, because so many times you come across something that you can't place, you turn to the literature doesn't really help either. It might not be in any of the books, and there comes a point where you kind of either throw up your hands or you go like, we're going to have to sequence it. And that's where sequencing comes in really handy. And then just sort of a final reason why sequencing can be a useful thing is, and this is a little rant that my friend Damon Tai posted on Instagram the other day, why the obsession with putting names to things, because that's what sequencing kind of helps you. Names help us see the diversity around us. If we can't see the diversity in our own neighborhoods and local ecosystems, we have little chance of being good stewards for that diversity. So sequencing helps us see and appreciate the diversity around us. And obviously conservation, fungal conservation is sort of a topic, I'm very passionate about. And a lot of fungal conservation is about documenting and verifying records of very rare fungi, often in threatened habitats. And if we are to get some of them red listed and protected, we need data. The biggest barrier between getting more protections for fungi is just the lack of data on distribution and incidence and all that. So having verified collections of rare species can bolster a case to protect a certain area. And the other thing is that we are losing fungi faster than we can actually document them because of habitat destruction, development, nitrogen accumulation, and so on. So documenting fungi before they disappear, before we're never going to see them again, using sequencing is sort of another sad but important thing, where sequencing comes to really good use. So those are all the whys of why it's important to sequence. It's about getting names, but it's about much more than getting names. But let's talk about DIY sequencing then, because I think a lot of people want to dip a toe into that water. Just very briefly, an overview of the different stages of the process. We're going to go through them in a little bit more detail later. The first is the extraction phase. The DNA sits within the nucleus of the cell, and it's sitting in there pretty tightly. DNA doesn't want to leave the cell. It's sort of, for obvious reasons, so it needs to be extracted. So that's the first step, getting the DNA out of the nucleus. The second step is, so you're going to have a few strands of DNA when you do that, but to do polymerase chain reaction, you need billions of strands of DNA. You need a lot. So you're going to have to amplify those few strands and turn them into lots. And that's the second stage. The third stage is optional. You don't have to do gel electrophoresis. I know it's a bit of a mouthful, but essentially what it is, it's a test where you test whether you actually amplified your DNA or whether you got a blank, which happens before you send it off. Because the last step in the process is unavoidably, you will have to send it to a lab for Sanger sequencing. Sanger sequencing is a Sanger sequencing sort of machine costs $60,000 and it's very finicky. So you definitely can't do that at home. But the good news is it's cheap and it's quick. It's not nearly as expensive or as time consuming as sending it off to Alva Lab or Molecular Solutions. So let's talk about those 4 steps in a little bit more detail. All of these can be done at home, and I'm going to talk about the equipment and the cost and all of that in just a minute. But the first step is extraction, extracting the DNA from the cell. So you take a tiny, tiny little piece of fungus. Doesn't matter if it's the cap or the gills or the stipe. Doesn't matter if it's dried or fresh. You take a tiny piece, smaller than a sort of grain of rice, and put it into a tube. And then you add, there's several different protocols. This is the protocol I use. You add some sodium hydroxide. Sodium hydroxide is very basic. It's essentially Drano. And it's a very harsh chemical, so it breaks down those cell walls. Then you grind the sample with a little pestle and turn it into sort of a soupy liquid. You add a little bit of buffer to dilute it, and then you heat it to 95 degrees. So you can see all the different sort of things that you're doing. You're using force, you're using chemicals, you're using heat to essentially sort of coax that DNA out of the nucleus. And then when you've done all that, you've cooked it for about 10 minutes in a thermocycler, you're going to put it into a centrifuge. And what happens there is sort of all the cell debris falls to the bottom of your tube and the DNA floats to the top. So that's your extraction. The next step is amplification, and this is where it gets a little bit technical, but I'm hoping to sort of explain it in a way that makes sense, because you're adding just a tiny little bit of that sort of DNA liquid that's floating at the top of your tube to a bunch of other things. You're going to add it to primers, I'm going to explain what that is. You're going to add it to some master mix with Taq polymerase and some water. So you're essentially just adding it to a mix of all these different things. And every single thing here has a role. And then you put the whole thing into a thermocycler. A thermocycler is a machine that heats and cools. That's literally all it does. And the first step of the process is called denaturation. And it's a fancy word for like, it's going to cook the DNA. And what happens when you cook the DNA at 95 degrees, the two strands of the DNA split into halves. So instead of one full strand, you have sort of two half strands. And then you cool it down. And you put a bunch of primers into the mix. And what primers are tiny little bits of DNA. Because what you're not trying to do is you're not trying to copy the whole string of DNA, all 47 million sequences. That would be crazy, because you're only interested in like a tiny little bit of it. So what a primer does, it attaches to the front and to the back of the ITS regions, the only thing you're interested in, and it attaches to the front and the back of that region. And then in the next phase, when you sort of heat it up a little bit again, the Taq polymerase essentially doubles that little area between the primers, suddenly kind of gets turned into a full strand when it was just a half strand. So essentially, you've just doubled the ITS region of the DNA. And you did it once, and you're going to do it again, and you're going to do it again. In fact, you're going to do it 35 times. So you're turning one strand of DNA into 2, into 4, into 8, into 16. And after 35 cycles, you're going to have several billion strands of DNA, which is pretty crazy. So that's what amplification does. So now you have several billion strands of DNA, and you're kind of done. You could just send it to the lab for Sanger sequencing now. But, you know, Sanger sequencing still costs a couple of bucks, and you don't want to send something off that might not have worked. Sequencing still costs a couple of bucks, and you don't want to send something off that might not have worked. So you're doing gel electrophoresis. And what's involved here is pretty simple. You're making a jelly block with agarose. It's just like a little block of jelly. You can kind of see it in the picture. And then you kind of, it's got little holes, little so-called wells at one end, and you submerge it in a little bit of buffer. And every single well, you just sort of inject a tiny little bit of your amplified DNA, and you put a stain on it. You can kind of see the stain here. So every well gets its own little sample, and then you turn the current on, and DNA wants to travel towards the positive end of the current. So it's starting to migrate through the gel, sort of like floating through that gel. And because you put a stain on it, it actually shows up under a special kind of light, like a blue light. And it looks like this. You can sort of see this kind of jelly block with the DNA in the wells. And you can see that most of the lanes have like a bright fluorescent band across it. And that means this the amplification was successful. But there's one kind of dummy down here. It doesn't light up. There's no fluorescence here. That means that particular sample was not a success. So I can save the $3 and not send it for Sanger sequencing. So it's an optional step, but it's pretty useful, especially when you're starting out and you still make mistakes. So the last step, Sanger sequencing, like I said, it requires an incredibly expensive machine and like experts in white coats. And to explain it is a little bit painful. And I'm not going to spend a ton of time on it, but basically they're doing PCR again, and they're doing gel electrophoresis again, except when they're doing PCR, there's a specially labeled fluorescently labeled oligonucleotides that cut off after a certain length. And then they get sorted into sizes between large fragments and small fragments, and then a laser beam gets shown through them, and then through a photo multiplier, and that eventually shows up as a letter code in a chromatogram. So it takes all the different bits of DNA that are floating about, and every single sort of base of that piece of DNA gets translated into one of those beautiful arcs, a G or an A or a C or a T. They all have different colors that correspond to them. just a little bit about the cost of Sanger sequencing, because obviously that's going to be a hot question. How much do I have to pay for this? And there's 3 suppliers that I know of. The lab that I use is called MC Lab in San Francisco. They charge $2.99 for a forward read, or $4.99 for forward and reverse. Forward and reverse is like best practice, because if one of the reads is a little bit iffy, the reverse kind of, you can overlay it and together they make a good read. But, if I'm feeling confident, I will often just do the forward and then see how that goes. So that's $2.99 per sequence. GeneWiz only do forward and reverse. They charge $6.30 for that. They're also very popular. They have, great customer service, and it's a little bit more of a full service experience. And then Eurofins really only becomes affordable when you buy a whole plate for 96 samples. So if you're sequencing for foray, or if you're sequencing your herbarium, it might make sense, because that breaks down to about $3.41 per sample. So those are the costs of sequencing. As you can see, it's a lot less than if you send it out to Alva Lab or one of the other vendors. And the turnaround time, I usually, if I in the woods on Sunday, do DNA sequencing on Sunday evening, pop it in the mail on Monday, I get my sequence results back usually first thing Monday morning. First thing Thursday morning. So it basically takes about 4 days to get the results, which is nice because everything's still fresh in your mind. The samples are still in the fridge. You can do some microscopy on it. So it's actually pretty nifty to have that quick. Super quick, one quick slide on what does the future look like. The future mostly looks like Oxford Nanopore, MinION, which I think is how it's pronounced. It's a tiny little gadget that you can hold in your hand, and it sequences DNA without amplification or Sanger sequencing, but you still need to do extraction. You can take it into the field, you can hook it up to your computer. It gets used a lot by scientists actually in the field. And there's a protein in it that pushes the DNA through a tiny little pore. It's a very, very small pore. They sort of apply an electric charge. You can measure that charge, and that's converted into a DNA base. So it's a much simpler, much more elegant system than all the other stuff that people are doing today. Cost is $1,000 for one of those units, but you're also going to have to buy flow cells, which is sort of the reagent part, and if you're going to use fewer than 100 species, if you're going to sort of sequence smaller amounts, you're going to have to use a fungal, which is sort of an adapter that allows you to sequence smaller amounts. But the net cost is about $1 a sequence if you don't count the cost of equipment. It used to be not very accurate. It's getting better. And if you're like, organizing a foray or have some other high volume project, it's definitely a tool to seriously look at. There's one member of our club who might be buying one, and I'm all ears. I'm kind of excited. Obviously, it's still far beyond a reach for most people financially, but things always get cheaper over time, and I'm hoping that this will happen to this. A lot of people are starting to experiment with it, so it could be really cool.
So let me first introduce myself. I'm Tom McCoy. I've had a lot of different positions in the club over the years. And most recently, I have become the nominal head, air quotes, of the DNA sequencing group. I say nominal because I am easily the least knowledgeable person on the sequencing team, but that is changing rapidly. So let me give a little bit of background on how we got to now. So DNA sequencing is something that we've been doing at MAWDC for several years. And in the first bullet, I say MAWDC and Microflora 1.0. Microflora was the name of the North American Mycological Association funded initiative for mushroom clubs to do DNA sequencing. This was rolled out several years ago, and MAWDC made a substantial investment in it to purchase vouchers to do sequencing. In this initiative, we didn't the clubs didn't do any of the lab work themselves. You simply dried the sample, and you got a grain-sized piece of tissue and sent it off to the lab so you could get your results set. The and it had, I think it had very great expectations, a really wide kind of idea of what they were going to be able to achieve. I'm not sure they achieved everything that they set out to, but they still sequenced a lot of material. And for our club specifically, I just sequenced the last vouchers that we had at our Sequinota foray last fall. It was at that time that I first got involved with the mycoflora, with the DNA sequencing group, and much to my surprise, I thought I was volunteering to lead a group. It would be a nice little easy, lazy work group, and I wouldn't have to do very much. But we've been going at a blistering pace ever since. And this is MAWDC and mycoflora 2.0. This is our current DNA sequencing team. And I've listed the team members here. There are a few other people who are involved. The biggest one of note, our science advisor, Dr. Megan Romberg. She's been just phenomenal, getting us up to speed. And we have a couple of really other talented people on our group. You heard from Annie before. She's obviously so knowledgeable and excited to learn. Anil Saranthu has a background in genetics. He's a great addition to the team. My good friend Serenella Linares is also on the team. She's all but dissertation in mycology, so she can talk at length about any of this stuff. And then I think everybody else is kind of at my level, which is kind of a base amateur coming into this. But you don't have to stay at that level for very long, because as I say over here, there's so much great content on the web for the autodidact. You know, we heard so much from Sigrid's presentation already tonight. Also in her references, she talks about the one by Damon Tai. That is a phenomenal presentation. It goes much more into depth. And she also made references to Alan Rockefeller. We heard from Alan Rockefeller at MAWDC last year. He is just in a limitless depth of mycological knowledge, and he has a lot of content out on the web too. So you can spend all day researching this to your heart's content. We have some goals, some tenuous goals that we've set for the current year, 2022. First of all, we need to buy the equipment to get set up to do the DNA extraction and amplification. That's all been completed. Great thanks to Megan Romberg again on that. Because of all of her years of experience, we were able to achieve that in short order. The other thing, and I am a particular stickler for this, we want to do good science, and good science means repeatable process. And that means we have to have solid documentation. So we're taking our time right now. But we're going to peer review this document, our protocols and our documentation many times. They're currently a work in progress, but a lot of them are getting to be in pretty good condition. And my goal is by the end of the year that we do have proper documentation in place. All this stuff will be made available on the MAWDC website for anybody who's interested in getting involved. That's a resource to see if this is something that you want to spend your time on. The other big goal is it's just not good for overall team health and longevity to have to depend so heavily on Megan's experience every time that we want to have a meeting. So we need to have numerous practice runs on executing the samples before the end of the year. We tend to do that about, well, what the goal is for the rest of the year is to do that about once a month. And so that'll give everybody a chance to get familiar with all the processing steps. This is not that difficult to do. You just have to go through a practice run a few times to kind of get your arms around what we need to achieve. I set a tentative goal for 100 samples this year to process. I think we could probably do a lot more than that, but that's not our focus. Our focus is to do quality work and to make sure that we have ample amount of time for the core team to all get up to the same level of standard. So if Megan, or if the more experienced people aren't available, other people can step right in with no loss of quality. And I touched on this up here. We purchased a lot of equipment to do this year. I am very cognizant of the fact that I am spending membership money, and the board was good enough to put this into this year's budget. And I want to make sure at the December meeting we give full transparency of what we've been able to achieve this year, including the result sets. So stay tuned. You'll hear from me again in December. Currently, we have a couple of, we have one project. This is going to be a longitudinal study at the Audubon Naturalist Society at Wood End. Probably a lot of people online are familiar with this facility. It is a really nice reserve in Chevy Chase, Maryland. And it's worth going to walk around over there, even if you aren't interested in picking fungi to sequence that day. We have an agreement with them. Serenella Linares, who I mentioned before, does a lot of the organizational work for them, and she's been great, instrumental for making this happen. It is really a great kind of proving ground for us to get up to speed. This also, and before the end of the year, we will definitely be doing, I would say, at least one run, probably 2 runs of DNA work at Sequinota. Sequinota is going to be four days this year, so that'll give us plenty of time to play around with doing extraction and amplification. We may be able to do some other MAWDC event. In my mind, I think it would be nice to have a DNA extraction kind of corner at the mushroom fair to give people a chance to get a little bit familiar with what this is all about, but we'll see. So, big question, why the heck are we doing this? I mean, this is a lot of work, and this is, a lot of new information to absorb. But I think the biggest thing, at least for me, citizen science is a force for good. I mean, I'm a computer programmer in my paying life, so I don't really have the background to do this, but if you put the time in, you can make a positive contribution. And whenever I say citizen science too, I'm mostly thinking of amateur mycologists, and just in my own experience, a lot of the best field mycologists that I've ever met were amateur mycologists. So we really should be leveraging that knowledge base to advance our overall knowledge. The advanced science, by having research grade observations, this is going back to doing good quality work. It's not about getting more observations done. The observations that we do, we want to make sure that they're well documented. It's almost impossible, to my mind, to have too much documentation. That's going to include things like spore prints and microscopy, and Whenever it comes to things like boletes, I think we should be talking about, how did they perform under chemical spot tests, et cetera. Like I said, you can almost not have too much documentation. And the final point is to have a herbarium. One of the other goals, if we're going to be advancing science, is we have to have this type of material available for the long time. There may be a researcher who shows up 5 years, 10 years down the road, having seen our INAT observations, and he may or she may want to go back and take another look at that sample. And we have to have that properly curated, so we can go fetch that material for the person. He or she may want to take a look at something outside of the ITS region. That's a very distinct possibility. So we want to make sure that we're doing good work for today, but for the future as well. The presentation just mentioned, discussing what's next. We just talked about next technology. This is the Nanopore MinION. I guess that's how you say that. I don't really see us doing anything with this for at least another year, maybe 2. And, as fast as technology moves, maybe that's a good thing. I am big time into lichens, so I cannot wait to add lichens to and potentially slime molds to the list of things that we're going to be doing DNA sequencing on. Lichen, pardon me. Lichens, though, that opens up a whole new can of worms. So it's a little bit different processing wise. You probably have to use, like a lot of lichenologists who are going to do DNA work, with lichens being a little bit tougher structure, it's a little bit more difficult to break them down to do the extraction. So I was just talking to James Lendemer about this, who is at the New York Botanical Garden. The way he breaks down his lichens for DNA extraction is he uses liquid nitrogen. So, that's another step beyond where we currently are, but we will get there at one point. I also think with lichens that will almost certainly have to be tied with better chemical analysis. When you're doing identification for lichens, the chemistry, it really drives everything. Lichens, because they live in such extreme environments around the world and have to tolerate, if you think of lichens up in the Arctic region, they're just blasted by ultraviolet light all day and freezing cold temperatures all night. And because of that, lichens have evolved more than 1,000 chemicals unique just to lichens. And a lot of that drives the identification. So it's not just DNA whenever it comes to identifying lichens, it's also the chemistry behind them. And a lot of that chemistry, you have to use thin layer chromatography. So anyhow, stay tuned. We'll be doing much more with these in the future. And of course, with our model, once we get the team fully comfortable and our protocols well documented, the sky's the limit with how much of this we want to do. Right now, we're really only committed to, like I said before, the longitudinal study at the Audubon Naturalist Society and a couple of our standard events throughout the year. But I can easily see us perhaps contacting the National Park Service to do something at maybe C&O Canal or Rock Creek Park, etc. So there's a lot of excitement behind what we're doing now, and I think that we'll be keeping our foot on the gas pedal for the next several years.
So you've sent your sequence to the lab, and what you get back on Thursday morning is a whole bunch of these things. They're called chromatograms, and they're very easy to read, very easy to open with this free software. I use one called SnapGene. And And you can see that there's a lot of sort of mess at the beginning over here. And there's a sort of like a slightly messy bit at the end. And you definitely don't want to copy that. That's like trash. What you do is copy all the good stuff, like this kind of, you end up with a sequence like this. So this is a Phaeolepiota aurea that I collected in Greenwood Cemetery, and I was kind of curious about how it fits. And then you go over to a website called Genbank. It has sort of an engine called BLAST. It stands for Basic Local Alignment Search Tool. Super easy to use, like a child can use it. It's Google for sequences. You pop in your sequences, you push the blue BLAST button, and it'll spit out something like this. So this is for my Phaeolepiota aurea. So the column you're most interested in is the column that says per ident, also percentage identity. That's really sort of where all the action is. And as you can see, there's a couple of sequences that have 100% match. I'm very familiar with these sequences. I put them in myself. So they're from Greenwood Cemetery as well. And I would be shocked if they weren't 100% matched. But that's where it gets interesting. The next closest sequence is 98.15, and they're all from Europe, different parts of Europe. And then further down, 97.53 from Washington, 97.62 from China. That's not close. This would give me pause and go like, are we even talking about the same species here? What I typically look for is like 99 and above in terms of percentages. So, okay. So I look at the Washington sequence and compare it with mine. So this is a direct comparison. The pink bits are where they diverge. And I'm counting like over 10 base pairs differences between the sequences. That's a lot. That's like 10 mutations, since they had a common ancestors. Okay. So next step is I build a tree. And that's very easy to do. There's a website called Phylogeny, where you literally just download a couple of sequences and pop them in, and it'll build a tree for you. And what we can see here is that like one branch of the tree is my New York City sequences up there on the top. And then on another branch is the Washington, China, and Europe sequences. They're not even on the same branch as mine, although sort of, like Europe all clustered together and China all clusters together. So the answer is it a new species? Is mine a different species from the other Phaeolepiota? Who's the true Phaeolepiota aurea? I don't know. We need to sequence more East Coast specimens to make sure mine's not just an outlier. And I'd love to get a mycologist on this because it's interesting. So that's what typically happens on your session with Genbank and BLAST. There's other databases as well. There's Unite, which is a European-based, more curated database that sometimes has unique sequences. There's Bold, which is based in Canada, that sometimes also has different sequences from Genbank. Micromap is almost like a meta database in that it will look at even things like sequences posted to iNaturalist that are not on Genbank. It's a little slower, but it's very comprehensive. And then there is MicroPortal, which is not really a place for analyzing sequences. However, it's fantastically useful for distribution data and other things that can support or answer questions about, is this, a new species, or, is this the same thing or is it different? So those are all great databases to know about and to use. Okay, so I said it's not all rainbows and butterflies. There's definitely issues with DNA barcoding, quite a few of them. A significant proportion of sequences in Genbank is mislabeled. Like somebody identified a fungus, thought they super confident, thought they're really right, popped the sequence into Genbank, turns out it's wrong. They might have even realized, but nobody ever goes back to correct. So there's just a lot of kind of misleading names in Genbank. Some people say it's as many as 40%. Many sequences will not have close matches. So you're getting stuff where the closest match is 95%. So you're just sort of on your own. You're not quite sure what you have. So you're going to have to do a lot of the detective work yourself. Not all species are easy to sequence, like carbonaceous paranomycetes, as we have discovered in our club, are pain. So you're going to have to culture them on agar and then sequence the mycelium. And the ITS region does not work for all species. And in fact, there's, you know, other regions you can sequence, and it's good practice if you're going to write a paper to do that. But, you know, this talk is not long enough to accommodate that detour. So it's definitely the beginning of a journey rather than the end. Oftentimes, a lot of extra work is involved, a lot of sort of like hitting the books and the microscopes and sweating. And one of the things I will say is, like, when you are looking at all the sequences that you find, a really important thing is to look who posted the sequence. Is it a trustworthy source? Is it a famous name? You know, if I'm looking for inosa-based sequences, I want to see the name Brandon Matheny. If I'm looking for waxicap sequences, I want to see the name Jean Lodge or somebody else who's really good with that genus, and so on. So I'm definitely always looking to compare to somebody who's a real authority in the genus, you know, not a grad student, you know, who might have just done this as part of their coursework. So it pays to dig a little deeper and not just trust the first sequence that seems close. Case in point, you know, this is an auricularia the club found on the club walk. It looked weird and a little bit different. We sequenced it, and, you know, the 100% match is the actual sequence. So no surprise that it's 100% match. But then you get auricularia cornea, auricularia delicata, polytricha, reticulata, and they're all sort of at similar levels of similarity. And when I click through to all of them, which I always do, is like, they're all Chinese sequences, which is not a bad thing. There's some amazing universities in China. I'm just not as familiar with some of the leading researchers there. So I can't necessarily say that this sequence is higher quality than that sequence because I'm familiar with the research or the researcher. So in this case, it's basically back to the microscope, because there's nothing here that gives me complete confidence that this is indeed auricularia cornea. Another thing that often happens is, you know, this is a russula I found in a local park. It's white and shiny. I went like, what is it? And Genbank tells me there's a whole bunch of sequences in Genbank that are russula species, several of them. But then the first named sequence is at 95%. So it's a russula, great. And then somebody threw in a trichomopsis decora, clearly mislabeled. You know, that's another thing that happens. So you're not necessarily any the wiser. You just know that other people have come across this very thing and they haven't figured out what to call it either. So it's sort of, you're throwing it back into the ocean, into sort of limbo. And, you know, and Stephen Russell, who's been very active on Genbank and North American Microflora Project, he actually started assigning code names to some of these undescribed, so at least we can begin to bucket them. This is an example of a russula that he calls russula species, Indiana 30. So it helps kind of bucket things if there were a researcher to ever want to investigate these. Just a few words on how to properly store something. If you were to give it to somebody to sequence, you know, keep your specimens away from other fungi, because contamination, make sure your tackle box is clean so there's no spores contaminating your sample. Don't let them sit in the fridge for too long. Don't let them go moldy, because then you get two sequences on top of each other, which is bad. Don't cook them, like don't dry them at too high of a temperature, 110 or below. And then keep the dried specimen in a Ziploc bag with a label that includes the INAT or MO number. Because, you know, once you get addicted, like I did, you're gonna, you know, dry more and more specimens. Like here, this is my little herbarium. Ideally, you would deposit your specimen in a proper herbarium, but a lot of them are not currently accepting submissions. They're understaffed and underfunded. So certainly make sure that it has a good home in your home, so that if a mycologist does come calling, you have a specimen in good condition.
Okay, that describes what you get back. And I post your questions into the chat and we will read those or invite you to ask your question. But the one thing that I wanted to start with on this really kind of goes back to the why are we doing this? What is it that we are developing? And my sense of it is that right now we're building out the database in Genbank, for example, so that when people plug in the information that they get from this sequencing, they will get matches to it, and they'll get well-identified matches. Sigrid, do you have some other comments about what it is we will ultimately accomplish by the citizen scientists gathering this information and putting it in?
Yes, absolutely. I first want to echo what you just said is like community science sequences are some of the highest quality sequences in Genbank. Because if you see a professional mycologist sequence, oftentimes all you get is a date maybe, and if you're lucky, a location. No pictures, no microscopy, no other characteristics. It's, you know, unless there's a published paper that goes with it. Whereas if you look at a community science, a sequence that has an INAT or MO record attached to it, you get all that stuff with it. So it's much more valuable and much higher quality, I would argue, than what the professionals do. So that's definitely, you're sort of furthering our collective knowledge about fungi, which is an amazing thing to do. You know, and in my club, obviously, we have also a really specific objective. We're trying to document the fungal diversity of New York City. And New York City is not necessarily a place that people think of when they think of fungi. But if we can prove that even in this very compromised environment, there's thousands and thousands of species growing, you know, that's interesting. You kind of wonder how these fungi adapt to living in such tough conditions and so on. So, you know, there's many different reasons to do this, but yeah, but, you know, furthering science is definitely a big one.
So when we plug that information into Genbank and then get a readout with a list of close matches. Just to be clear, that's just close matches for that small that ITS segment that we looked at. It's a little unclear to me exactly what it is that's important about the ITS region that's being sliced out of the full genome. And I was reading, listening to another conversation about this that suggested they're getting to the point where it's going to be inexpensive enough to do full genomes on fungi. And I was just wondering whether the ITS, why it is that the ITS region is what we focus on here. Was it just a matter of cost, or is there something special about that?
It's so much more than, actually, it's nothing to do with cost whatsoever. It's just a really excellent region in that it's highly mutated because it's completely irrelevant to the organism. You know, it plays no role in whether this organism thrives or dies. So it accumulates mutations without any punishment. And those mutations correspond almost directly to how different or how similar two different species of fungi are. And there's really no other region that's quite as good as doing that. There are other important regions that you can look at in terms of, you know, comparing fungi, but ITS is just a brilliant region. And frankly, for most fungi, you'd be wasting your money if you did full genome sequencing, because a lot of the information would be absolutely useless. You know, you would get the whole phone book when you really just want one page of the phone book. So yeah, that's the long answer.
So I'm going to chime in
Yeah.
just real quick on that. And Sigrid's right. The ITS is fantastic because the mutations, but it's also tied to a highly conserved area. So it's tied to the gene regions that encode for proteins that are involved in the protein mechanism, the ribosomal proteins. So you have these highly conserved that are going to be the same across genera, across families. So you have anchor points that you can use. And then between those anchor points, you've got this area that mutates quite a bit. So it's kind of a unique region in that you're not just looking at conserved proteins that are going to have very few changes, but you've got the very conserved parts and the mutated parts, and you have them together in one area and can compare across species and genera.
So when we get a readout from Genbank that says this is 99% similar, it sounds like most people view that as, well, those are the same species, even though there are some differences. Can you talk a little bit about what we think the distinction is between a species when we get these readouts and things that we call species complexes?
I'm happy to take that. I mean, first of all, it differs quite a bit. For Cordinarius, for example, I would want like there to be no more than maybe two base pair difference or half a percentage point of difference between, because the species are very close together in genera like Cordinarius. With other species, that's not quite the case. When does it become a species complex? Probably, I want to say below 99%. Maybe around 98% or so, but I'd love to hear, Megan, you probably have a more informed perspective on how to delineate, but I think it's more of an art than a science, is my impression.
Right, and generally, you know, I mean, a blast search gives you that information. I can share my screen with a few things. That number is percent identity across the entire species. So it includes an area that's very conserved in the center, the 5.8 region. So that's going to be 100% often between groups. And then you've got the ITS. Generally, people consider about 97% for genus. So you want things to be about 97% similar to be in the same genus. And then between species up to 99, like Sigrid said. But when you're, you know, the blast search is one answer. If you really want to kind of answer, is this species or part of the species complex, you have to do the phylogenetic tree like Sigrid showed, and you have to align all of your sequences and really do some statistical analyses to see how they match up.
The phylogenetic tree is what is produced by SNAP gene, is what you're using, Sigrid?
No. I use the SnapGene is literally just a program for reading and fixing chromatograms. I use phylogeny, which is a website, which is very easy to use. You can also do phylogenetic trees straight off BLAST. And there's some very nice programs that cost money on which you can do them as well, but I'm poor. And, you know, so I use the free resources, but I'm sure Megan has some fine recommendations on what the professionals do.
Well, and I use Mega, which is free. It's a free software that you can download. And it's pretty easy to use, pretty intuitive. And you can look at trace files in that, and you can annotate them and edit them, and then you can align your sequences with things that you get out of Genbank or wherever.
Annie, if you unmute
yeah.
yourself, you should be able to talk.
Thanks. Yeah, I just wanted to bring up and kind of get people's thoughts on it. But one of the things that came up in a talk that a bunch of us, I think, were listening to last week was that percentage, you know, it's like based on a different kind of baseline size of the areas you're comparing. So you could be saying 99% of, you know, a really not very great 50 base pair read, or you could have 5 different genes included in that percentage. And so the denominator is different, and that may affect what the percentage means. So I don't know if that's getting too granular, but that was a really mind-blowing thought for me.
Damon Tilge's discussion seemed to suggest that the base pair length of the ITS region we're talking about is maybe 250 to 500, 600 base pairs. And so when you're talking about percentage, is that what you're focusing on, Sigrid?
Honestly, you don't always, I mean, so it can be up to 700 base pairs, but sometimes you get a crummy sequence and there's only 300 base pairs. And obviously that impacts, you know, like Annie was saying, you might think it's a great fit, but you're not looking at the full picture. So yeah, it gets pretty subtle. Not every sequence is.
Oh, I see. When you put in your list of base pairs, it's only going to match that size piece in what's in Genbank, and so you may not be matching against the whole ITS region of other things that have been sequenced. Right,
but it can still be valuable and useful. It's a little bit different.
Yeah, like maybe you could get lucky and get 50 really important base pairs and catch some differences, but there is a, like, I think, isn't it true that with some type species, like they'll try to sequence it, but they're so old that they've already been broken up and maybe you can only get like a short sequence out of that type sometimes?
That can happen. Do you mind, could I share my screen? See all of these colors?
Yes.
All right, so this is an alignment of different sequences. So this is from the Coprinopsis. And as you go along, so this is the ITS 1 and 2 and the part in the middle, the 5.8. So you can see, like, right in here, these are all pretty much identical. So this is the 5.8S region that's between ITS 1 and 2. So if you happen to only submit this section, you might get 100% match to all of these different sequences. The ITS 1 is over here, and you can see all the white spots and all the different colors, and that shows the mutations. And on the other side of it is the ITS 2, and you can also see the white areas and the different colors. And so what you want is something that covers all the way from ITS 1 across to ITS 2 and lets you really know what your percent match. And you're going to have sort of an artifact from the central part because it's going to match across a lot of species. And you're looking for these single differences that occur in the ITS regions. So you do need to be careful what you're comparing with what.
So does that suggest, because you've got a lot of Straussmerii on the left-hand side there where they're not identical in some of these regions, that they're mislabeled in Genbank?
So mislabeled. So this is the name that was put in them in Genbank. And the other thing I just want to kind of show here real quick to keep in mind is this is a visual I like to show people. So on the left, this is how many fungal species we estimate might occur in the world. This is how many species have been described, and this is the taxa in Genbank. So we can put in sequences and maybe get a match to what's there, but there are a lot of things that are described that don't have sequences, and then there are a whole lot of things out there that haven't been described or sequenced. So just because you've got a name over here, that's the name that whoever was looking at put on it as a best educated guess. It doesn't necessarily mean that is truly Straussmerii or truly variegata or whatever. So those names are whatever the person in Genbank put on as the name.
Mm-hmm. Right. Are you able to, if you get this kind of readout, are you able to make your own guess as to whether that seemed to be a reasonable name to apply to it?
Well, then what you get after that alignment when you do the phylogenetic tree could be something like this, where in pink was our sequence from Maryland, and that matches up to some other sequences from parts of North America. But you have all of these other buckets, as Sigrid calls them, all these other groups. And so we're putting names on something based on morphological characters that when you look at the underlying genetics, there's more going on. And these will probably need new names eventually.
Mm-hmm. We're also talking about the difficulty of getting a good sequence, and there's this the one of the important steps is doing the gel run after you've extracted the DNA. And Megan, I know you're working a lot on protocols as to how to do this process. Could you talk a little bit about what it is that can go wrong that makes the gel sequence reveal that you didn't accomplish what you wanted to accomplish?
Sigrid, do you want to talk about what everything can go wrong?
Oh,
There's
sure.
so many things.
I mean, somebody was texting me their gel yesterday because I was lending out I lend out the lab to club members, and sometimes things go wrong. So she texted me a gel and was completely blank. And it turned out that she had used 2 forward primers and no reverse primer for the amplification, but it could have been other things. It could have been Like the thermocycler wasn't switched on, or it could have been she forgot to put the stain into the gel, or so I have a whole troubleshooting section in my protocol to help people figure out what exactly went wrong, because there's a whole bunch of things. But she did it again, and it came out good.
Yeah, and there's so much that can, even you can do everything right. You can add everything perfectly right and still not get amplification. Sometimes there are inhibitors in your sample that don't work. Sometimes your primers need to be at a higher temperature or a lower temperature in order to anneal. PCR can be pretty tricky. And so even if you've got really great DNA and you've done everything right, it can still fail. And you just try it again, and you maybe lower the temperature, or you do the DNA extraction again. Maybe it was a little dirty, maybe you had inhibitors. So PCR can fail regularly.
It sounded like one of the things you were working on, Megan, with our DNA group was that there would be other regions or other primers to use. Can you talk a little bit about what that is and why we would be doing that?
Well, so the ITS is the group of great regions of the barcode that everyone agreed on, and you got the mutations there. But sometimes you don't resolve species. So a lot of the things that I work with professionally, you'll put in an ITS sequence and it'll be 99.9 to 100% to all of the described species. And so you need to look for other gene regions to actually be able to find differences between species. So people have looked at a lot of other conserved proteins. They're generally involved in cell building or some other conserved process. Beta tubulin, which is a protein that's involved in the skeleton structure of the cell, the elongation factor that's involved in cell division. And so, nowadays, a lot of people will look at up to 10 sequences and then jump to the whole genome sequencing. So it would depend on if there's a particular group, like if we decide we want to focus on Russellas or chanterelles or whatever, you would look into the literature and see what other gene regions people have used so that you could compare yours to what other people have done. But it's generally other proteins that are conserved. There's nothing quite like the ITS.
Mm-hmm. Okay. Well, that raises the other question. Sigrid, I'm curious, how do you decide what your club is going to sequence?
Yes, you need like a special kind of radar. You look at all the fungi on the table, and some of them you just, something's off about them, or something you think you know what it is, but something's not quite right. So, you pick the weirdos and the ones that kind of trigger your kind of curiosity. But and oftentimes you're right, but sometimes it ends up being something really common, but even that is valuable. But I want to say we have a pretty good instinct for what's going to yield an interesting result. But yeah, so it's not a scientific process.
It's the oddity of the morphological difference that you're looking at in the first instance, as opposed to deciding you're going out to look for a bunch of things that are the same to find out whether they have different sequences.
Right. Like most often it's like we're looking at something weird, and we've tried to idea it, and we've come up short. But for example, I'm really interested in Inocybaceae, and I will go out and look for Inocybaceae and bring back like 20 and try and figure them out, obviously morphologically first, but then if I don't succeed, definitely sequence them, which is almost all of the time.
It sounds like the end result is that once you get this information, like the example you gave in your discussion, Sigrid, that, well, we looked at all these things that we thought they were pretty much the same and found out that they had different DNA sequences. And then you go back to the microscope and to examine, okay, well, how are these things different? But you've got to have good samples of what it was that you had examined and you had picked in the first instance in order to be able to go back to the morphology again, so people can identify them without the DNA. Is that really what that process is?
Yes, you should not be sequenced the same as a species twice. once you've sequenced it, you should know it like, it's a member of your family. You should study it and really immerse yourself in that species so that when you come across it again, you won't need DNA sequencing, because otherwise you're just putting a lot of money in the meter to spit out a name over and over and over again, which seems wasteful and, not what we community scientists do.
Mhm. So you also are pretty active in fundus, and I know fundus is pivoted to preservation of red list fungi. And my read on that was that, well, it was because it was too difficult to build out all the fungi in the United States, and they needed to narrow their focus. But the idea that we're losing fungi as a result of environmental change strikes me as, given the small number of the whole fungi that we know of, how can we really say that, as opposed to, oh, the environment's degrading and so there's a lot more fungi rotting stuff now, so we have more fungi.
I wish that was the case. Yeah, no, we just know from overall environmental assessments that, the world is losing species diversity. Like I recently heard the number of 1 million species have recently been lost, and that's obviously not just fungi, it's whole ecosystems, because it's not just like when you lose ecosystems, you lose the animals, you lose the plants, you lose the fungi as well, and that's just what's happening everywhere through development and nitrogen deposition and climate change and whatnot. It's not like we can prove that we're losing tons of fungi, but it's just what's happening overall, and fungi are so clearly bundled in with all those other ecosystems. It's happening, and their distribution's changing and everything else. But yeah, and just on fundus, the reason they might well come back to sequencing. It's just a case of funding and manpower, the sort of pivot away from sequencing. And if we can find more grants, and we might well come back to doing that, because obviously it's a really valuable thing to do.
There was a question Elizabeth raised here, which is, said that the Chicago club did a deep dive on chanterelles and ended up finding undescribed species that had been lumped together. What is this lumping and splitting issue for the DNA sequencers?
Right. I mean, well, you're touching on two things. First of all, like obviously when you do sequencing, you find tons and tons of undescribed species. I mean, I found like dozens of them. I mean, anybody who sequences will find undescribed species left, right, and center, because like just having looked at that chart that we saw earlier, there's just so many fungi out there that have, that have not been described yet. In terms of lumping and splitting, I mean, that's obviously a philosophical debate. It's very easy to use DNA sequencing to split. And because a lot of academics depend on having papers out there with their name on it, it's very tempting to do a lot of splitting, because that allows you to describe new species and accumulate sort of academic credentials. There's not so much lumping going on because, there's no money in it. But whether something should be lumped or split, it's as much of an art as a science. Does it have real utility? Will it help? Are there actually real differences instead of, some, like tiny little differences that nobody in their right mind would ever notice? So that's a whole other discussion. But yes, sequencing sometimes gets used for splitting and not always, in ways that make sense.
Is lumping for species and splitting is for genera? Is that what's happening here? Is it because we seem to get a lot more name changes at the genera level than the species level?
Yes, splitting is definitely happening mostly for people taking a genus and then just actually taking the species and then saying it's not just one species, it's actually 3 or 4 species, like the example I was giving you with Elsa, taking the one species and kind of splitting it. Yeah, I can't answer that. Maybe Megan has a better answer. I'm punting.
Well, so I mean, taxonomists, there have always been lumpers and splitters, even when it was just based on morphology. There have always been people that thought these characters are actually meaningful to split these things apart, or these characters are not meaningful. So this is just sort of another iteration of that. And there are some cases like in a fungus called alternaria, which is a little microfungus that's super common. You've probably all eaten it without knowing it because it's all over all sorts of fruits and vegetables and everything. There were about 40 species that have all been lumped together in the same species now, based on DNA sequencing. So, you know, back then, sort of everyone on a new host was given a different name. So it's in mycology especially, we have such a lack of characters, you know, compared to entomology and some other things. You just don't have that many characters. With mushrooms, you have more. With the microfungi, you have very few. And so finding those differences and our technology and microscopes haven't always been adequate to finding those differences. So when you have 600 base pairs, those are basically 600 characters that you can look at. And using all of those, you can figure out how things are related. So the splitting is in many ways more just a reflection of increased knowledge.
Mhm. Annie, you had a question about the Northeast rare fungi. You want to follow up on that? I don't know very much about it myself.
I
Sure.
can.
Yeah, I would agree. I was hoping Sigrid could just like say what it is, that
Mhm.
with challenges, and I'm actually really hopeful that our club might be able to find a couple of things. Like there's one, so if maybe she can say it, and then I'll say what my wish list is.
Right. Okay. So this is a fundus initiative and started on the West Coast. We kicked it off a year and a half ago on the West Coast because like the most important thing anyone can do for fungal conservation is actually just doing observations of really rare fungi. Basically, keep doing what you're doing, just do, and you guys are already doing it, because that data is really valuable. Fundus funnels it directly to a company called NatureServe, which then funnels it to land managers and so on, and they can use it for protection programs and other initiatives to sort of protect habitats that harbor rare species. So in order to encourage people to document more rare species, they've got this great challenge. And it's essentially 20 species that have been selected that we need much more data for. And the challenge hasn't been launched yet. It'll get launched, I want to say, in the next 3 weeks. It'll be 20 species. It has boletes on it. It has ascomycetes on it. It's got squamanida and all kinds of other really cool, easy to recognize fungi. You don't need a microscope for those. And it's going to run for like 3 years. And we just want to sort of see which of these people will find. Maybe some of them will turn out to be really common. Some of them we'll never find. But it's going to be a lot of fun. I ran the West Coast one, and it was just so cool to see, you know, what people were actually finding. So what's on your bucket list, Ali? What do you want to find?
Oh, I'm so excited. So I'm hoping we might be able to find, like, I'm probably saying it wrong, but Winia sparasoides, or Winia, I don't know which way you say it.
Yeah.
Winia, because I saw that there have only been a few recently. I guess there are a couple more observations, but in the past, one of them was in the 80s, and it was from Maryland, and it looked like it was like about 30 or 50 miles west of Baltimore. So maybe around the Frederick kind of area. So I was super hopeful that we might be able to just try to like follow that path and look at the parks that are there. I mean, that was a long time ago. Maybe, you know, there's been a lot of development, but I was just hopeful maybe we can find a kind of like micro habitat and maybe find that one.
Right, and they'll all get sequenced. That's part of the program. We had 10 rare species on the West Coast. It turns out it's now 12 rare species because 2 of them turned out to be new species that people had found as part of the challenge. So yeah.
Yeah, I heard you went to the West Coast for one trip and found one of them immediately.
Yeah, which actually has now changed the whole species concept because my family managed to get a sequence out of it. So yeah.
But like, I love that you literally like set foot on the West Coast and we're like, oh, there's a rare species. Okay.
No, that was lucky.
But it's fun.
It looks like we're we're not
Yeah.
going to have time to run the description, which I think is very interesting, of the things the New York Mycological Society has found in its DNA testing. But I would encourage people to take a look at that. We had on the poll, about 70% had not looked at the video at all. And so you've missed that relatively interesting and important part. Though it doesn't tell you how to do it, it certainly shows the sorts of things that you may turn up. Megan, I understand that our preliminary results had turned up some what we thought were unusual things as well. Can you describe what was going on there?
Man, I just closed that. You were closing down. It was the tree that I showed previously with the Coprinopsis that you know, there's diversity there that hasn't been recognized based on the morphology. And so that was just that just happened to be the fungus that Annie brought to our initial DNA extraction party, just as something for us to practice. And so,
But it was,
you know,
I had collected it for the reason Sigrid said, which is that there was something weird about it, because it looked like Coprinopsis variegata based on all macro features, except it had these rhizomorphs that were feet long. Like, I have pictures of it, like, and it's supposed to be Strauss-Mayeri, or however you say it, that's supposed to be associated with the rhizomorph. So there was like, it was one that I saved for a reason, because it was a weirdo.
yeah.
Great. Anybody else had some burning questions that they want to ask? Otherwise, we can just ask Megan later. Our homegrown expert.
Megan, could you talk a little bit about if there's anyone still on this call that's interested in getting involved in what you're doing? What's the best way to tap in at this point?
I'll let Tom answer that question.
Okay.
I'll post my email in the chat if anybody's interested. Please just reach out to me. We'll be very happy to add more people to the group. I think or just ask me questions in general. I think whenever it comes to this type of work, it's already been elucidated pretty well tonight. It's not like going on a normal foray where you just toss stuff into the basket. You're going to spend 20 minutes photo documenting and setting up an INAT observation, writing notes in the field. So, you know, this is what you're in for if you want to do this. But yes, we're absolutely looking for more people. So let me put my email in the chat.
And also, if there's limits to how many people can learn the process right now, you can still save stuff you find this summer. So if you find anything cool this summer, save it. And it's easy to dry it at 105 degrees or lower, you know, not too hot like you would dry something to cook. And, you know, put it on INAT. There is a field you can add for whether a specimen is saved or not. So, you know, that's a nice field to add. But you can be part of the project even now, just going out on your own.
And also, if you're interested in doing this and you haven't watched any of those reference videos yet, that's probably your first step. Go check out Sigrid's video. Check out that one by Damon Tai, and after you watch those, you'll know, you'll really have your arms around what this is all about.
If you dry or freeze something that you think is interesting, waiting around for the opportunity to do the sequencing, how long reasonably might you preserve that before you won't be able to get DNA out of it?
Well, somebody just did a project where they sequenced PEX type specimens for hebelomas, and they were 120 years old or something, and they got good DNA out of quite a few of them. So your answer's right there. If you preserve them carefully, potentially quite a long time. Freezing is a new one. I bet it works, but it's not normally how people do it. So I recommend drying on that.
Well, thank you, Sigrid. I really appreciate you putting together the presentation as well as joining us to field some more questions on it.
Oh my God, this was so much fun. This is my passion. Thank you for having me.
Great. Okay. Anything else, Elizabeth, before we close down?
That's all we got. Thanks, everybody, for joining us. This was great.
Okay, bye.
Okay, bye.
Bye.